Good afternoon, everybody. Today's learning objective is about Introduction to Proteins and Amino Acids. Organic compounds which are containing one amino group and one carboxyl group in the same molecule are known as the amino acids. And the amino acids on combination, they give the peptides, polypeptides. Polypeptides on combination, you get the proteins. So 20% of the human body, it is made up of the proteins. Proteins are the large complex molecules that are critical for normal functioning of cells. They are essential for the structure, function, and regulation of body's tissues and organs. So 20% of the human body, it is made up of the proteins, and the proteins, they have various functions, large complex molecules which are critical and normal functioning of the cells. They are essential for the structure, function, regulation of the body tissues, defense mechanism, and building for the organs. They are the building blocks for the body. Proteins are made up of smaller units called amino acids. These are the building blocks of proteins, and proteins are the building block for the body. They are attached to one another by peptide bonds, forming a long chain of proteins. Now we'll see amino acid structure, its classification. An amino acid contains both carboxylic group and an amino group. Amino acids that contain an amino group bonded directly to alpha carbon are referred as alpha amino acids. Every alpha amino acid has a carbon atom called an alpha carbon, and this is bonded to the carboxylic acid group. So the COOH group, an amino group is bond NH2 group, a hydrogen atom, and an R group that is unique for every amino acid. So the carbon valency, it is tetravalent. One will be bonded to the amino group, one to the acid group, one to the R group, and one to the H. Here you can see the carbon. This is the amino group. This is the COH group. This is the R group and H. Now we'll see the classification of amino acids. There are 20 amino acids based on the nature of the R group, and they are classified based on their polarity. So amino acids, if they have hydrophobic amino acids, they can be so the amino acid, how it is classified, we'll see. Carbon atom, it is having tetravalency. One is bonded to the amino group, NH2 group, and one bond is to the carboxylic group, and one bond is to the R group, and the next one, the alpha carbon bond, is to the H. Because the COOH group is the main functional group here, and the carbon attached to the COOH group is called as the alpha carbon. Now we'll see the classification. Classification of amino acids. Amino acids, there are 20 amino acids based on the nature of their R group. They are classified based on their polarity. Amino acids are hydrophobic amino acids, and amino acids can be hydrophilic amino acids. So hydrophobic amino acids are the nonpolar amino acids. R is the alkyl group and R can be the aromatic group. If the hydrophobic group, the alkyl group, as it changes, the different amino acids are glycine, alanine, valine, leucine, isoleucine, methionine, proline. And if the R group is an aromatic group, the examples are phenylalanine, tryptophan. Now coming to the next type of amino acids, hydrophilic amino acids, polar amino acids. These are again classified into three types: neutral amino acids, acidic amino acids, basic amino acids. In the neutral amino acids, we can see tyrosine, serine, threonine, cysteine, glutamine, asparagine. These are all the neutral amino acids. The number of acid groups are equal to the number of basic groups. These are called as the neutral amino acids. And the polar amino acids, the next category is acidic amino acids. In the acidic amino acids, we have two acid groups rather than the amino group. The acidic groups are more, so the amino acid is the acidic amino acid. It is glutamic acid, example, aspartic acid. And coming to the next one, the polar hydrophilic amino acids, the third category is basic amino acids. In these basic amino acids, we have more number of basic groups rather than the acidic group. So yeah, you have even the acid group, but the number of basic groups are more. And in this basic amino acids, lysine, histidine, arginine, these have more number of NH2 groups. Still, the amino acids can be classified further based on the position of the NH2 group from COOH. Alpha amino acids. So these amino acids, in the alpha amino acids, position of the acid group is present on the alpha carbon atom. Example, glycine, alanine, alpha amino acids. And the next category is beta amino acids. Thus, amino acid, in these amino acids, position of amino group is present on beta carbon atom. It is on the beta carbon atom. Next, for that example is beta amino propionic acid. We can take an example. And the third category of the is alpha amino acid, beta amino acid, and gamma amino acid. In this gamma amino acids, the position of the amino group is present on the gamma carbon atom. It is on the gamma carbon atom. So you take CH2, CH2, and for the COOH is having 3 carbons, alpha, beta, and gamma. And to that gamma carbon, we get the NH2 group bonded. Example, gamma amino butyric acid. And the next one is delta amino acid. These amino acids position on the amino graph and the amino group, it is on the delta carbon atom. So for the COOH group, you have 4 carbons attached: alpha carbon, beta carbon, gamma carbon, and delta carbon. To that delta carbon, the amino group is attached. So this is delta amino acid. We have seen the classification of the amino acids based on the polarity and non-polarity. And we have seen the classification of the amino acids based on the position of NH2 group from COOH group. Now, let us go for the next classification. The classification based on essentiality. Essential amino acids are the amino acids which you need through your diet because your diet cannot make them, are called as the essential amino acids. Actually, we are having 25 amino acids. Nearly there are 25 amino acids. And on hydrolysis of the proteins, you get this 25 amino acids. Out of these, 2 amino acids are present, proline, hydroxyproline. And remaining 23 are amino acids. Now in that, we have to see the essential amino acids. The amino acids which are not synthesized in our body, and hence they have to be supplied through the food for normal growth of the body. Such type of amino acids are called essential amino acids. Such type of amino acids are called as the essential amino acids. You have 10 essential amino acids: histidine, isoleucine, leucine, methionine, phenylalanine, tryptophan, valine, lysine, arginine. Okay, these are the examples. Lysine and arginine are not indicated here when they are the essential amino acids. Total 10 essential amino acids are there, which are required along with the diet. Now, what are non-essential amino acids? The amino acids which are synthesized in the body, and which are not essential part of our diet because they are synthesized by our body. Such type of amino acids are called non-essential amino acids. Such type of amino acids are called as the non-essential amino acids. In these non-essential amino acids, remaining 15 out of 25, 10 are essential and 15 are non-essential. So, alanine, arginine, asparagine, aspartate, cysteine, glutamic acid, glycine, ornithine, proline, serine, tyrosine. All these come under the non-essential amino acids. Now, coming to the next one, peptide bonds. And before going to the peptide bonds, we'll see amino acids. Another classification is there. Already I have told you, basing on the acidic nature, basic nature is one classification. Based on the essentiality is another classification. Based on the position of NH2 group is another classification. Next, there are different methods for synthesis of the amino acids. The different methods for synthesis of amino acids are from halogenated carboxylic acids. They can be prepared. How you prepare is carboxylic acids on treating with Cl2, Br2 in presence of red phosphorus, that is the HVZ reaction. It forms alpha halo acids, and further treatment with NH3 will give you alpha amino acids. And the next one is malonic ester synthesis. Malonic ester synthesis Malonic ester synthesis we treat with sodium ethoxide to form malonic ester. On treating with sodium ethoxide forms sodium malonic ester. This one, which on treatment with alkyl halide, it will give alkyl malonic ester. This one on hydrolysis, it will give alkyl malonic acid. So this alkyl malonic acid on halogenation and heating gives a alpha halo acid. This alpha halo acid on treatment with NH3, it will form alpha amino acid. So this is the way we prepare alpha amino acids using the malonic ester synthesis. Even using the Strecker synthesis, we can prepare amino acids, alpha amino acids. Aldehydes on treating with HCN form cyanohydrins. The cyanohydrin on treating with NH3 gives an alpha amino cyanide, which on hydrolysis forms alpha amino acids, which on hydrolysis will form alpha amino acids. So this is the Strecker synthesis, the most important synthesis. Aldehydes on treating with HCN form cyanohydrin, and the cyanohydrin on reacting with NH3 gives an alpha amino acid, alpha amino cyanide, which on hydrolysis forms alpha amino acids. Now let us have a look on to the physical properties. A melting point, both acidic and basic amino acids, they are, as the salts, have high melting points. Coming to the solubility, they are soluble in water. So, coming to the physical properties, we have seen the solubility. Now coming to the optical activity. Amino acids even exhibit optical activity. And the next one is amino acids exist as Zwitter ions. Amino acids exist as the Zwitter ions. Now, that is a dipolar ion. They exist as the Zwitter ion. Sorry, was. Now coming to the Zwitter ion, dipolar ion. In aqueous solution, amino acids, in aqueous solution, the amino acids, acidic carboxyl group can lose a proton and the basic amino group can gain a proton. And this kind, the internal acid-base reaction takes place and a dipolar ion is formed. This is called as the Zwitter ion. Now, the properties of the Zwitter ion which amino acid exhibits is amino acids are crystalline solids having high melting boiling points. This favors Zwitter ion formation. Amino acids have high dipole moments. Amino acids are water soluble and insoluble in organic solvents. And amino acids does not indicate, the IR spectrum of the amino acids does not indicate presence of NH2 and COOH. So these are the evidence in favor of the Zwitter ion. Now coming to the peptide bonds. So before telling this peptide bonds also, amino acids have amphoteric character. They have isoelectric point. Each amino acids is having the different isoelectric point, isoelectric pH. The pH of the amino acid solution at which amino acid does not migrate to any electrode under the electric current is called isoelectric point. And coming to the chemical properties, amino acids, the amino group is having different chemical properties and the acid group of the amino acid is having different chemical properties. They undergo alkylation, acylation, react with the nitrous acid, nitrosyl chloride, they undergo deamination, esterification, ammonia, they react, decarboxylation, reduction, action of heat, all this can be observed, the chemical properties for the amino acids. Now we'll see what is a peptide bond. So the smallest these amino acids on combination will give you the peptide bond. So what is a peptide bond? Amino acids are linked together by amide group is called a peptide bond. During protein synthesis, the carboxyl group of amino acid at the end of the growing polypeptide chain reacts with the amino group of the incoming amino acid and releases a molecule of water. The resulting bond between the amino acids is peptide bond. The resulting bond between the amino acid is the peptide. Here you can see this one. This is the peptide bond. Here the water molecule is lost from this OH and NH2. This, all of them combined, water molecule is lost and a peptide bond is formed. So during protein synthesis, this takes place, a peptide bond. A dipeptide is formed between two amino acids. Tripeptide, it is formed between three amino acids. Polypeptide, it is formed between three. Tripeptide, it is means three amino acids are formed. Dipeptide means two amino acids. Tripeptide means three amino acids. And a polypeptide means n number of amino acids will be there. And the nomenclature of peptides we'll see. For example, before going to the structure of the proteins, what is the nomenclature of the proteins? Glycyl alanine. A glycine and alanine will join to give glycyl alanine. Okay, now in peptides, the amino acid part which contains free amino group is called N-terminal acid, and the amino acid part which contains COOH group is called C-terminal acid. In peptides, N-terminal is always written on the left side and C-terminal acid is written on the right side or the polypeptide chain. Now let us see the structure of proteins. To determine the structure of proteins, to determine the structure of a peptide, it is complete hydrolysis should be carried out. So peptide is refluxed with dilute HCl so that the peptide linkages are broken. Analysis of resulting solution gives us the kind of the amino acid amounts present in the peptide. Now structure of amino acids we'll see. The sequence of protein, it can be determined by DNA of the gene that encodes the protein or that encodes the portion of the protein for multiple unit proteins. A change in gene's DNA sequence may lead to the change in the amino acid sequence of the protein. Even changing just one amino acid in a protein's sequence can affect the protein's overall structure and function. To understand how a protein gets its final shape or conformation, we need to understand the 4 levels of protein structure. That is primary structure, secondary structure, tertiary and quaternary structure. Now we'll see in detail what is a primary structure. The simplest level of protein structure, primary structure, is simply the sequence of amino acids in a polypeptide chain. The hormone insulin has 2 polypeptide chains, A and B. The sequence of A chain and the sequence of B chain can be considered as an example for primary structure. Protein structure is complex. The protein structure is made of 3 steps. Sequence of amino acids in protein indicates Sequence of amino acids in protein indicates primary structure. The amino acids are arranged in regular sequence which repeats itself. You can see here. They get repeated for themselves. Alpha helical structure, and now thereon the primary structure gives us the secondary structure. The secondary structure refers to local folded structures that form within the polypeptide due to interaction between atoms. The most common types of secondary structures are the alpha helix and beta pleated sheet. Both structures are held in the shape by hydrogen bonds, which form between carbonyl oxygen of one amino acid and the amino hydrogen of another amino acid. Next, coming to the here, two structures of the secondary structure. It is having two structures. Before going to the tertiary, in detail, I'm going to explain what is a beta pleated and what is the alpha helix. First, we'll see alpha helix. Polypeptide chain is arranged in a spiral structure. It is called as alpha helix structure. The coiling or the spiral structure of polypeptide chain is formed by intramolecular, you can see it in the molecule, intramolecular hydrogen bonding between NH and CO group of the same chain. The alpha helical structure we can see, and intramolecular hydrogen bonding we can see. Now going to the secondary beta pleated structure. This is formed by the side-by-side arrangement of polypeptide chains which are stabilized by intermolecular hydrogen bonding between bond NH of one chain and CO group of another chain. So the intermolecular hydrogen bonding between NH group of one chain and CO group of other chain. Intermolecular hydrogen bonding can be observed here. And coming to the next one, the tertiary structure. The tertiary structure is the three-dimensional shape of a protein arises from further folding of a polypeptide chain to give a complex structure. So the three-dimensional shape of a protein arises from further folding of the polypeptide chain to give the complex structure. We can see the tertiary structure. The overall three-dimensional structure of a polypeptide is called tertiary structure. The tertiary structure is primarily due to interactions between the R group of the amino acids that make the protein. Importantly to tertiary structure, there are the hydrophobic interactions in which amino acids with nonpolar, hydrophobic R group clusters together on the inside of the protein and leaving the hydrophilic amino acids on. So after the secondary structure, the tertiary structure is formed. I've told you the three-dimensional. The overall three-dimensional structure of a polypeptide is called the tertiary structure. The tertiary structure is primarily due to interactions. The overall three-dimensional structure of a polypeptide is called its tertiary structure. The tertiary structure is primarily due to interactions between the R group of the amino acid that makes the protein. Importantly, tertiary structure of the hydrophobic interactions in which amino acids with nonpolar, hydrophobic R groups cluster together on inside of the protein and they leave the hydrophilic amino acids onto the outside to interact with the surrounding water molecules. Also, the disulfide bonds covalent linkages between the sulfur containing side chains of cysteines are much stronger than the other types of bonds. You can see here. So the three-dimensional arrangement you get. The secondary structure, beta alpha helical, then beta helical, they get three-dimensionally arranged and they form here, where you have the disulfide bonds, covalent linkages, sulfide side chains, cysteines, and these are more stronger structure will be formed for proteins. And finally is the quaternary structure, where multiple polypeptide chain subunits come together when the protein attains its quaternary structure. Example for the quaternary structure is hemoglobin. Hemoglobin carries oxygen in blood and it is made up of 4 subunits, 2 each of alpha and 2 beta. So this is the structure of the proteins. You can see the folding. So first the primary structure, the second one is the secondary structure, and the third one is the tertiary, and fourth one is the quaternary structure. And what are the functions of a proteins? You know, proteins play a major role in our human body. The functions of the protein are, they act as the carriers, they have the defense mechanism. The blood, it carries oxygen, so carries the food particles for the drug transport, for the defense mechanism. So in that, it's the building block for the body.